I am Shamba Saha, and I'll tell you about our work on intercellular positioning of liquid-like organelles. The nematode worms C. elegans begin as single-cell embryos, which undergo a series of unequal cell divisions to generate precursors for different tissues in the adult organism. During these divisions, a class of cytoplasmic organelles called P granules are segregated only to the precursors that eventually give rise to sperm and eggs. Correct P granule segregation is thought to be important for fertility in worms. An interesting feature about the P granule organelles is that they do not have a lipid membrane separating them from the surrounding cytoplasm. Instead, like oil and water separate from each other, several proteins and RNAs simply separate from the cytoplasm to form the P granule phase. These P granule components exchange constantly with the cytoplasm. We discovered that of the 40 or so proteins in P granules, only one protein, PGL3, can work together with mRNA to make a P granule-like compartment in a test tube. P granule formation is concentration dependent, such that if the local concentration of PGL3 or mRNA is reduced sufficiently, P granules dissolve. P granules are initially distributed all over the one-cell embryo, but segregate to one end before cell division. How does this work? A clue came from earlier studies which showed that P granules segregate in response to signals from the cell polarity system. These signals generate concentration gradients of mRNA binding proteins like MEX5. We found MEX5 can bind to most mRNA molecules in the cell and does so more strongly than PGL3. Where MEX5 levels are high, MEX5 takes away mRNA from PGL3 and P granules dissolve. But on the other side of the embryo, where MEX5 concentration is low, P granules persist and grow. So dissolution on one side and growth on the other side leads to P granule segregation. Also, we were able to recreate P granule segregation in a theoretical physics model that used parameters we measured inside cells. There are many other important RNA protein compartments in cells, and the RNA competition mechanism we have identified here could represent a powerful mechanism of organizing the spatial distribution of these compartments inside cells.